Okay, so hi everyone. Today we're going to talk about autoimmune conditions and how, once again, hypersensitivity situations with our immune system being sort of overreactive and making mistakes in improperly identifying certain types of signals in the body as being threats can actually lead to very serious problems. Right? So when we were talking about the issues that our immune system presents to make it difficult to transplant things like blood or solid tissues, we were talking about how the immune system, right, is built up to be very sensitive to detect things that do not look like self-signals. Right? When we talked about blood, we said that the ABO and RH antigens on the surfaces of blood cells are very, very immunogenic. So the immune system can pick up on those very quickly and easily, and that can elicit a very strong response, oftentimes leading to the rejection of donated blood or donated tissues within minutes or hours of the botched transplant. We said that also T cells can be involved. If T cells ultimately identify wrong signals, for example, the MHC shapes on donated tissues, because the donor's MHC, their polymorphisms, their profile that they inherited from their parents, if that's not very similar to the HLA profile of the patient or the recipient, then you're going to have the patient's T cells potentially bind to the differently shaped MHC platforms on the otherwise perfectly healthy, normal donated tissue, and that can cause rejection. Right? What about this notion of stem cell transplants? So I'm sure all of you have heard about stem cell transplants. What is that? Right? So when you have a situation where the immune system itself may be the cause of a cancer, because remember that B cells, T cells, they have nuclei just like most other cells in your body, and therefore they have the potential to actually become cancerous if these cells start becoming out of control and proliferating. So oftentimes, how would you actually deal with clearing the body of an immune system that itself is the source of disease and cancer? So that's where stem cell transplants come in. So the idea behind a stem cell transplant is to essentially replace a person's immune system that might have, problematic B cells, for example, with new stem cells, hematopoietic stem cells from a donor's body that don't have these genetic mutations that give rise to problems. So when we talk about bone marrow transplants, that is essentially what we are getting at, the transplanting of stem cells. Now, as it turns out, when I was younger, I thought a bone marrow transplant was literally you're transplanting the marrow of bones from a donor into the recipient's bone, right? But that's not really how it works. Right? Stem cells are harvested from the donor, right? And these can come from the donor actually having bone marrow, right, removed through things like a needle or something. It's not a very pleasant experience because they actually have to get into the bone marrow to get those hematopoietic stem cells out, right? Then they are kind of cleaned up and purified, and then they are given via IV infusion into the bloodstream of the recipient. And these hematopoietic stem cells then will flow through the blood, and eventually many of them will end up just in blood vessels that lead to the small little capillaries in the bone marrow. and then they will exit the blood into the bone marrow, and they will hopefully repopulate the bone marrow with this different version of stem cells that come from the donor. So in order to do that, I think it seems pretty clear that you need to first clear the patient's body, the recipient's body, of their existing stem cells. So you essentially have to erase or ablate the existing immune system to make way to receive new stem cells. So if you look at this cartoon here and this slide, so normally the donors who give bone marrow are healthy and alive, right? The patient who's going to receive these stem cells, they have to receive some pretty high doses of cytotoxic drugs and radiation to essentially destroy their existing bone marrow and essentially destroy all of their own hematopoietic stem cells in their bone marrow that they were born with. So we call this myeloablative therapy. So ablate means to destroy, and in this case, we're destroying the stem cell in the bone marrow. By getting rid of the existing immune system, then you avoid, or hopefully avoid, any conflict between the patient's existing immune system, right, and the types of signals that it looks out for, and suddenly these brand new stem cells that are generating a whole new immune system, what signals they look for. And of course, by ablating the patient's immune system, you make room, physical room, inside the bone marrow. So here you see in the cartoon, the patient who's going to receive the transplant, they receive radiation and cytotoxic drugs, right? So this is a very scary thing if you think about it, because this person presumably is already very sick, right? With some kind of hematopoietic cancer, for example, like a leukemia, right? And here you are erasing their immune system to make them essentially as vulnerable as possible, right? So then they receive the IV transfusion of the hematopoietic stem cells that were harvested from the donor. Hopefully, over time, these stem cells will repopulate the ablated bone marrow in the patient. And you can see, hopefully, then these stem cells will begin to multiply and begin to regenerate, right, all the immune cells that are needed to essentially reboot or restart the immune system, but importantly, with somebody else's immune system, right, in terms of their stem cells coming from somebody else's body. So when this happens, it usually takes about 2 to 3 weeks for granulocytes and natural killer cells to start appearing again. B cells, T cells, so these small lymphocytes take longer to restore because remember, we learned about how B cells and T cells, how they develop through this whole process of having to undergo VDJ recombination, right, and selection, tolerance, training. And remember, the T cells, when the precursor cells leave the bone marrow, they have to first go to the thymus and go through that extra process of being told to become T cells and develop T cell receptors. So during these several weeks following a hematopoietic stem cell transplantation, the patient remains very vulnerable, right? And they are closely monitored in the hospital to try to make sure that they don't end up suffering from any kind of opportunistic infections. So if you're going to do a stem cell transplant, it's very important then to try to match the HLA types, the HLA profiles that the patient has from their mom and dad, and the donor, what they received from their mom and dad. Because remember, the shapes of the MHC platforms end up being a very immunogenic target for the immune system to potentially have issues with, right? So consider this. In this cartoon here, you have thymocytes that were derived from a donor's tissue, right? The donor's stem cells, and then this is now in the cartoon, this is taking place inside of the recipient's thymus, right? So in the patient's thymus, you will have the donor-derived thymocytes undergoing this process of being encouraged to become T cells. And of course, part of them becoming T cells is their brand new T cell receptors have to work against MHC, right? So these T cells that now develop, right, that are rebooting in the patient's body, they get properly trained against the patient's own MHC shapes, right? So in other words, they learn to recognize the patient's HLA profile and their MHC shapes. So these circulating T cells then, right, when they go to work, you would expect that they should function pretty normally in the patient's body because they have been selected for so that they know how to interact with the MHC1s and MHC2s of the patient's own cells that are presenting various peptides for these T cells to check out. But here's a problem. The other types of immune cells that are repopulated from the donor's stem cells, right? Things like macrophages, things like dendritic cells. What MHC shape will they have on their surface to do their presenting? They will still have the donor's MHC profile, right? Because they are immune cells that were derived from the donor stem cells, right? And so when they form dendritic cells, monocytes, macrophages, right, the MHC platforms that they have will be the donor's shape. As a result, if these shapes are too different from each other, you might actually have a situation where even though in the patient it looks like the immune system has successfully rebooted and repopulated, you will have brand new T cells that know how to interact with the MHC shape of the recipient's body, and yet they cannot interact with other immune cells like dendritic cells and macrophages because those are using the donor's MHC shape, right? So as a result, you will miss out on a lot of the interactions that are necessary between things like the helper T cells and other immune cells. And so as a result, it may be a situation where the adaptive immune response in the recovering patient is just nowhere near where it should be, right? So that's if the donor and recipient HLA profiles are too different from each other and they were not properly matched up when trying to find a suitable donor for this patient. On the other hand, there are some interesting situations where intentionally having the HLA be a little bit mismatched can actually serve a purpose. By having the HLAs a little bit mismatched, as the patient is regenerating their immune system using stem cells from the donor and different immune cells like natural killer cells that repopulate from the donor stem cells, right? They know to look for things like different types of shapes like MHCs from the donor's cells, right? They can actually be used to recognize maybe, for example, some leftover cancer cells that the patient may still have following their original, for example, leukemia treatment, right? So when a leukemia treatment is treated with different types of drugs, I should mention that a hematopoietic stem cell transplant is not the first line treatment for leukemia because of all the risks involved, right? The whole risk involved in eliminating the patient's old immune system in an attempt to reboot it. I mean, that is very risky, right? So most of the time, leukemia patients, right, will first receive different types of chemotherapies, different types of drug treatments just to try to eliminate the cancerous cells first. And that oftentimes can be largely successful, but there's always going to be some leftover cancer cells, right? And you worry about those leftover cancer cells potentially causing a relapse, right? So if those patients then undergo a stem cell transplant, perhaps the oncologists or the physicians, when they're trying to match up donor and patient, right, rather than trying to match the HLA types too perfectly, they may actually want to see if they can have a little bit of difference between the HLA types so that the rebooted immune cells can actually recognize any leftover cancerous immune cells as being different because of different HLA types. So in this case, what we're saying is, if you look at this cartoon, if you have a recipient, right, the person who needs the transplant, and let's say they have two different sets of polymorphisms, HLA polymorphisms, one inherited from their mom, one inherited from their dad, right? That's what's represented here. Yeah, these bars represent the chunk of DNA on the chromosome that contain all the HLA genes, right? So what they're saying here is maybe this particular chunk, right, has these three different HLA genes on it. This is where the HLA-A gene is, the HLA-B, the HLA-C gene. Remember, they're highly polymorphic. So maybe from their dad, let's say, they got this green set of polymorphisms. And from their mom, they got this pink set of polymorphisms, right? So ultimately, the HLA shapes in their body, right, is a combination of shapes due to these different genetic variations that they got from mom and dad, right? And that's what makes them unique. Right? So above it, let's say that the oncologists who are treating this patient have identified a suitable donor. Now, you might say, well, the best scenario would be if the donor has green and pink set of genetic variations to really match the recipients, right? Because then you feel like, oh, there shouldn't be any problem with compatibility. But what they may do is actually choose this donor here. This donor has a green set, just like the recipient has a green set, but this donor has a slightly different set. That's a light blue set, right? That way, if you look in the patient's body, when the stem cells that are transplanted over from the donor, so these are stem cells that know how to do the blue and green shaped MHC platforms, right? These cells, as they develop, right, and they repopulate the immune system, you'll have natural killer cells that come from these, right, that are essentially trained to kind of recognize that blue and green are the right shapes, right, to be a functional part of this rebooted immune system, right? But if you then encounter inside the patient some leftover leukemia cells, those leftover leukemia cells would have the green and pink shaped MHC platforms, right? Because these were the shapes that the patient's original immune system had, because this is what they were born with. Therefore, in this case, the rebooted or repopulated NK cells may actually recognize these leftover cancer cells as being a different shape. And therefore, you might encourage those natural killer cells in the rebooted immune system to attack and eliminate any residual or leftover leukemia cells, right? And so this kind of an approach is called graft versus leukemia, or GVL. Essentially, the grafted or the transplanted immune cells, you're counting on them to kind of seek out and attack any leftover leukemia cells that the patient still has. All right, so let's move on then to our discussion of autoimmunity, right? So to get onto the same wavelength here, let's remember, right, that all of our discussions about how the adaptive immune system recognizes different signals as being okay to tolerate them versus being different, and therefore signals that the immune system should be alerted about, right? Because maybe it's a threat. This all depends on being able to tell the difference between self versus non-self signals. Right? So that's why we emphasize that our whole adaptive immune system functions because it has to be self-tolerant, right? So that your B cells and T cells, and for that matter, your NK cells even, don't attack normal healthy cells that are you, right? So every day, our body's tissues present our immune system with far more healthy self-antigens that our immune system encounters, right, compared to pathogenic antigens. Right? So here, this table just reminds us that some of the mechanisms that contribute to self-tolerance are things like, for example, in the process of developing B cells have to go tolerance training in the bone marrow as well as in the blood, right? And any developing B cells that do interact with self signals have to be eliminated, right? They cannot be allowed to go to work. Otherwise, that will cause trouble. In the developing T cells that are in the thymus, remember, they have to undergo tolerance training as well. So the thymic epithelial cells, right, present all kinds of peptides to the developing T cells, again, as a negative selection process to make sure that any new T cells that, by bad luck, interact with self-peptides, right, that are presented by the thymic epithelial cells, that those potential problem-causing T cells are eliminated and not allowed to go to work. Right, so that's also what we're saying here, negative selection of T cells in the thymus. One thing that we didn't really talk about was that the localization, or where things like B cells and T cells go in the body, right? It's not like they are spread evenly throughout all the tissues in our body. I mean, you already kind of know that, but one thing that's worth mentioning is there are certain tissues where you really do not want to risk those tissues being the site of some sort of adaptive immune response that might lead to inflammation, right? Sensitive tissues like in the eyes or the reproductive organs, or of course the brain, right? You don't want to have an inflammatory immune response in the brain. So therefore, those are examples of tissues where lymphocytes are discouraged from hanging out where they're going. Another point, of course, is that any autoreactive B cells and T cells that are in the circulation, right, as part of their tolerance training, they're told to go anergic, right? They're told to shut down, right, so that they don't cause problems. Oh, and lastly, we did mention there's that special subgroup of CD4 helper T cells that are the regulatory or Treg CD4 types, right? And their job is to actually suppress or discourage fellow T cells from activating, right? And that keeps the noise level, the background leakiness of our adaptive immune response from getting in the way of our everyday lives. Right? So all of this is to emphasize again that self-tolerance is a critical given kind of part of an adaptive immune system, because otherwise you and I would always have reactions every day, right? If our adaptive immune system was allowed to react against signals that are our normal body's tissue signals. So that's what autoimmunity is then. Autoimmunity is a failure of self-tolerance. Right? So importantly, let's make a distinction here between the term autoimmunity and immunodeficiencies, which we were talking about before. Right? Immunodeficiencies, what we were talking about before, is that when you have a immune system that is missing something, right? Either due to an inherited genetic variation in a gene that causes the protein product of that gene to not function well, or maybe you don't even get a protein product from that gene, and depending upon how important that protein product is in your immune system, right? It could be a very minor immunodeficiency or a very major immunodeficiency. Likewise, we said that immunodeficiencies can actually occur later in life People who are born with immunodeficiencies, people who are born with a fully healthy, intact immune system might be exposed later in life to maybe a workplace chemical or toxin or something, or even a pathogen like HIV that would cause some component of their immune system to then be compromised, right? So in contrast, autoimmunity is different than that. Autoimmunity actually assumes that the person was born with a perfectly healthy immune system, right? And has maintained a perfectly healthy, robust immune system up until the point that something happens to cause that immune system to now turn on the body itself, right? And I want you to consider, if we routinely generated immunoglobulins or T cell receptors against our own body's antigens, then our health would always be compromised, right? If we didn't have adequate self-tolerance, you'd always be living with your adaptive immune system fighting against the tissues of your own body, right? So obviously we could not evolve that way, right? So think about autoimmunity then as like a civil war that's taking place, right, between two sides of your body, the immune system and your own body's healthy tissues. And just like a civil war, things can be very messy, very complicated, and it can be very hard to determine what exactly triggered this situation to unfold, right? So to date, we know of over 100 different autoimmune diseases, similar to things like allergies. There is the very noticeable and curious observation that autoimmune diseases are more common in affluent, industrialized countries. And again, right, even though we know much about how many of these autoimmune diseases cause problems in the body, there's still very little understanding of how self-tolerance is actually lost, right, to actually cause these autoimmune conditions. We do know that it's likely a combination. There is certainly a genetic component to it, certainly a component of developmental and environmental factors. And yes, right, pathogens can play a role, right? So we'll take a look at some examples of how pathogens, we now know, can often be a strong causal link with developing autoimmune conditions. So I said that there's around 100 or so different autoimmune diseases. This table does a nice job of just showing, a small assortment, 10 or so examples of well-known, well-studied autoimmune diseases. And what you see is these autoimmune diseases can be broken down into 3 categories, right? And once again, this type of condition fits with a hypersensitivity model, right? So again, remember, hypersensitivity means your immune system has become overly sensitive and starts detecting some sort of signal that in reality it should not be concerned about, right? So we looked at hypersensitivities as the reasons we have allergies. Hypersensitivity models are the reasons why tissue transplantation and organ transplantation is such a big deal, so difficult, right, to trick the patient's immune system to not be hypersensitive against donated tissues. And now, once again, we see that hypersensitivity, right, is the sort of background sort of condition for autoimmune diseases as well. So here in this table, we see that autoimmune diseases fit type 2, type 3, and type 4 hypersensitivity models, right? Just like tissue transplantation. The first 3 over here involve antibodies that end up being developed and produced that by the patient's body that unfortunately, right, end up detecting the patient's own self signals. Type 3 involves antibodies, maybe binding with other types of signals in the body that end up resulting in immune complexes forming, right? These large immune complexes that can't be cleared by the body, thereby causing chronic or long-term decline in health. And type 4 hypersensitivities that don't involve antibodies, they involve T cells making mistakes, right? So again, one way of thinking about it is allergies, tissue graft transplantations, and autoimmune diseases, right, all involve hypersensitivities. That means they all involve mistakes that the immune system is making against different signals. In allergies, those signals are harmless, maybe environmental signals, right, like pollen, dust, peanut, shellfish, right, animal dander. In graft and transplantation hypersensitivities, right, those signals are healthy, normal signals of things like MHC shapes that simply just don't come from the patient's own body, right? They came from somebody else's body. Now, for autoimmune diseases, we say once again, mistakes are being made where the patient's immune system are now detecting signals that should be left alone, and instead they are reacting against them. But here is the scary part, right? These are not external signals. These are not signals coming from the environment. These are not signals coming from donated tissue that's put into the body. These signals are the patient's body itself, right? So that right there makes this a very serious condition to treat. All right, so what we'll do is we'll take a look at these 10 examples on this chart here, but today let's just start off with these three, one from each category. Autoimmune hemolytic anemia, which is a type 2, systemic lupus, erythematosus, that's a type 3, and multiple sclerosis, MS, that's type 4. All right, so let's start with this type 2 example, autoimmune anemia. So in this particular condition, the patient's developing B cells, some of them end up coming up with IG shapes on their surface that end up being the shape of a person's own, for example, blood type, right? Or other types of highly immunogenic shaped molecules that are on their surface of things like their red blood cells, right? So of course, normally, such B cells that are developing in the bone marrow, right? I mean, obviously, every day you will have among the hundreds of millions of B cells that are developing in your bone marrow, some of them will just come up with IG shape recipes based on VDJ recombination that happen to be a shape that would match with, let's say, your own ABO blood type or other types of antigens on your blood cells. And that happens, but it's okay because what? As part of central tolerance training, those developing B cells are eliminated, so they never go to work in your immune system and cause trouble. But something may happen to cause a mistake. And if these naive B cells, right, that have IG shapes that bind to, for example, your own red blood cells, are allowed to pass tolerance training and allowed to mature and go to work, then you got a big problem, right? Because right away, these B cells are going to be selected for and chosen because they're going to be binding, right? Their IGs are going to bind to the abundant red blood cells in your body. And then if these B cells are told to become plasma cells to release antibodies, then you have big trouble, right? So in the case of these patients, their own antibodies end up attacking their own red blood cells. And therefore, their immune system begins to eat up their red blood cells, right? Phagocytic destruction by macrophages that are basically using these antibody stems as a signal that they should be eating these red blood cells. The antibodies binding on these red blood cells will cause complement fixation, right? That's the classic complement pathway. And then, of course, the macrophages CR1 complement receptors will then also pick up on that, right? And of course, antibodies themselves, as part of humoral immunity, the antibodies themselves opsonizing the red blood cell will cause things like membrane attack complexes to form and red blood cells to lyse. Right. So that's, I think, pretty straightforward to understand, that if you end up with B cells that fail tolerance training improperly, in other words, they end up passing tolerance training when they shouldn't, right? If they have certain Ig shapes that happen to be shapes that are abundant in your own self tissues, you could have a big problem. All right. Lupus is an example of type 3 autoimmune disease. So this is a chronic disease. So lupus happens when IgGs are improperly made against a variety of different cellular proteins. So not just necessarily your red blood cells, right, but other solid tissues. And so what happens is you'll get immune complexes forming between these IgGs and antigens that are abundant in the body, and you'll have the cross-linking of these, and you'd actually have these clumps, right, of these immune complexes of antibody and antigen, and you'll have so much of them that your body cannot clear them from your system, and they can do things like end up getting stuck in your blood vessels, like in capillaries. So that can lead to things like heart attack, stroke, or organ failure if the blood supply is cut off to the tissues of an organ. So this is the first time I'll mention this, but this is a very curious fact about autoimmune diseases. 90% of lupus patients are women, right? And not necessarily old women. You know, these can actually be quite young women, women between the ages of 15 and 44 years old, right? So it's true that lupus patients, women outnumber men, right? Almost 9-1, right? So there's currently no cure for lupus, but we can treat symptoms with anti-inflammatory drugs to keep the inflammation down, right? And if needed, then patients, you know, can undergo organ transplants, right, to do things like hopefully give them a new liver or a new kidney, for example, right, that may have been damaged by these immune complexes, right? So the symbol for lupus is the purple butterfly, and that has to do with the fact that one of the symptoms of lupus is this facial rash, right, due to this inflammatory response caused by these antibody complexes, right? So in recent years, certain celebrities, like this is Selena down here, right, have made public's awareness of lupus much more widespread, right? Okay. So the example of a type 4 autoimmune disease is multiple sclerosis, or MS. I'm sure many of you have heard of MS, but maybe you don't really know what it is. MS has to do with the nerve cells that transmit signals from our central nervous system out to our muscles. So nerve cells, I'm sure you've all seen before a diagram of a nerve cell. A nerve cell has a main body to it, and then it has these long axons, right, that are essentially like the long cables that connect nerve cells with other nerve cells, or nerve cells with, let's say, muscle cells, right? So just like transmission lines, these axons are actually surrounded by insulation. You know, just like the electrical wires in your house, right? Your electrical wires, the actual metal wires inside of them are wrapped around sort of these rubber types of insulation. Not only does that prevent you from getting shocked if you touch the electrical wire, but also insulation speeds up the efficiency of signals going through. So let's look at this. This image with the kangaroo shows that a normally insulated axon, right, as shown here, these sort of sausage-looking things are these special myelin sheath cells, right, that are wrapped around your axons, and they provide insulation. A properly insulated axon will allow a nerve impulse to travel very quickly, maybe on the order of 400 kilometers per hour, right, to get from, let's say, your brain to the bottom of your spine, and then from your bottom of your spine, maybe down to the muscles of your foot, right? It's very fast. In fact, if you want to think about wiggling your big toe, and you decide to do it, it seems instantaneous, right? You want to move your toe and it moves, right? However, if you have destruction of these myelin sheath cells, as shown with the snail here, right, then you're losing the insulation, and that can dramatically slow down the efficiency by a fold of maybe even like 100 or so slower, right? So literally, this would be a delay or a lag from the time you decide you want to wiggle your big toe to your big toe actually responding, right? So how does this happen, and why is this autoimmune? Well, as it turns out, in people who have multiple sclerosis, their CD4 cells, right, you have a population of them whose T cell receptor shape improperly recognizes peptides that are presented by these myelin sheath cells that surround axons. So obviously, these CD4 T cells should never have been allowed to go to work. They should have been identified and eliminated in the thymus, right? But now that they are allowed to go to work, they are attacking the nerve cells. They attack the nerve cells and they release things like interferon gamma. And so the interferon gamma recruits macrophages. The macrophages come to where the helper T cells are, and the macrophages begin to eat away at the myelin sheath, right? So the symptoms here are loss of sensory skills, muscle weakness, right? Because you can't even maintain normal muscle tone all the time to be able to stand up still, and loss of motor function, right? Coordination is lost. Again, this condition affects relatively young people, people between the age of 20 and 50, right? That's normally when the onset of MS is seen. And again, right, this is more frequent in women than men, about twice as likely for an MS patient to be a woman compared to an MS patient being a man. Okay. So before I go into this slide, then, I'm sure many of you are wondering, well, how is it? What's the explanation for why autoimmune diseases are more prominent in women than men, right? Now, for decades, people have tried to get to the source of this. Now, you would think the obvious thing would just be to think about what types of hormonal signals in a woman's body might be different than a man's. And you might then think about, oh, is it something to do with estrogens versus testosterone? But they've already ruled those things out a long time ago, right? Those very obvious types of approaches for looking for differences. What they do think now, right, is that it does have to do with the woman's immune system, right? The female immune system having to have evolved to be able to tolerate things like pregnancy, right, and being able to carry, right, a developing baby inside of them, whose genetic makeup is technically different than the woman, right? Because the baby's genetic makeup is a combination of the woman's and her partner's, right? So I think some of the prevailing kind of hypotheses are that it has something to do with the way the female body's immune system, the way the whole tolerance training works to allow for things like pregnancy. Maybe that somehow sets the stage for more mistakes potentially to be made, right? Okay, so when you have T cells involved in autoimmune disease, of course, that also means that the T cell was not properly identified in the thymus, right? Not properly eliminated. Now that brings us back then to the importance of the thymus as being the main self-tolerance training site for developing T cells before they're released. Remember, the way that the thymic epithelial tissues select for T cells, right, to be eliminated is by presenting all kinds of peptides that normally you wouldn't think should be expressed in the thymus. Right? These are peptides that might be expressed in other places in the body, but the thymus has a special enzyme known as the AIR, right? That the AIR is the transcription factor that goes around in thymic epithelial cells and turns on at random different expression of genes. So you have all kinds of peptides that are expressed in order for the developing T cells to train against, right? And hopefully the developing T cells will have seen a very, very thorough, wide representation of self-peptides that they might encounter in the body. And therefore, if any of those developing T cells react to those in the thymus, they are eliminated, right? So what if you have a situation where people are born with genetic variations in that AIR transcription factor itself, right, that prevent the thymic epithelial cells from properly displaying all kinds of peptides to train T cells, right? So that can definitely lead to autoimmune disease. So autoimmune polyglandular disease, or APD, results from the body's lack of properly tolerance training T cells in the thymus before letting them go, right? So people who have this inherited disease, their allele for their AIR gene, right, it's defective. So therefore, they cannot properly tolerance train their T cells, and T cells are allowed to go to work that end up detecting self signals. All right, so you can see here, this is some data from a European study where many of these types of autoimmune sensitivities end up resulting in different tissues that are autocrine or hormonal in nature being targeted, right? Like the thyroid, the adrenal glands, reproductive tissues like the ovary or testes, etc. Right? There are other types of tissues as well in the body that are affected, but in each case, this has to do with T cells that should have been eliminated in the thymus because their T cell receptor, right, happens to bind to peptides that are presented by normal cells in the body. But because in these people, their thymuses weren't doing the proper job of being able to train these T cells, that leads to these conditions. All right. So I said that autoimmune diseases, then, very clearly, there's a strong genetic component behind autoimmune diseases. Right? We know that. So what are the genetics of autoimmune diseases other than the one I just described, where if you have a defect in tolerance training, yeah, that can lead to B cells, T cells that should not have been allowed to go to work. Those end up going to work and causing autoreactive problems, right? But what about other genetic conditions, right, predispositions? Well, not surprisingly, much of the genetic contribution that has to do with autoimmune diseases has to do with HLA polymorphisms. Why? Remember, our adaptive immune system relies very heavily on cells like T cells interacting with our MHC platforms that all of the cells in our body that have nuclei, right, present on their surface as a way of properly communicating with the immune system, hopefully normally showing that healthy cells are healthy, right, and there's no strange foreign peptides in them, right? How do T cells see that? They see it because the T cells interact with the MHC platforms on the target cell and are able to read any of the little peptides that are being presented, right? So not surprisingly then, different shaped MHCs that people get because they inherited different HLA polymorphisms from their parents. Once again, it means that certain HLA profiles, right, the cells of the body of people who have those HLA polymorphisms, the shapes of their MHC platforms do a better or worse job of presenting certain shaped peptides. Right? And if you can then imagine that if there are certain peptides that are the types of signals that T cells are particularly sensitive to detecting, right, to give rise to autoimmune disease, then people whose cells present those peptides more abundantly, right, are at higher risk of their immune system seeing those peptides and therefore making that mistake, compared to somebody whose inherited HLA polymorphisms results in MHC shapes that are not good at presenting that particular peptide, right? Because in their body, if their cells don't even present that peptide, then their T cells are never going to see it. So their risk of their T cells making a mistake that could lead to autoimmune disease is much lower, right? So if we look at these, we've seen this cartoon numerous times before, right? So what we're saying is some of our HLA genes are highly polymorphic. Remember, like the HLA A, B, C ones, there are 1,500 or so variations in the general public, right? Whereas some of our HLA polymorphisms, right, are not that variable. But even then, remember, I said that even if there's something like 1,500 different cataloged genetic variations in the human population for something like the HLA gene, the reality is the vast majority of humans, right, only have maybe one of three most popular variations. So if you look here, that's what these pie charts show. We've seen these types of pie charts before, but we're coming back to it now. We looked at them before when we were talking about how NK cells have different receptors on their NK cells that help NK cells sort of check out different cells in the body to see if there's anything wrong with them, right? And remember, we said certain NK receptors are designed to bind to different HLA types as a way of kind of reading if a target cell is making normal amounts of a particular MHC on their surface, right? So now we're looking at these pie charts again, but in the context of autoimmune diseases. But again, we're saying that even for an HLA gene like HLA type A, where there's like 1,500 different variations in the public, the majority of people have maybe one of these three most common types, right? And then maybe the purple one here would be the fourth most common type. And then literally like another 1,400 or so other variations would all fit within the blue, the orange, and teeny tiny little slices you don't even see here, right? So remember that these variations, your cells express these different shaped HLAs on their surface so that your cells can have a better time more successfully. All the parts of the cartoon chicken, right? If you go back to that cartoon of the chicken being like some sort of a pathogen, right? And a pathogen protein, for example, right? And that protein is going to get processed by the cell into chicken parts. Remember, there's like a drumstick, a leg, a beak, a wing, etc., right? And we said that each one of those represents a small little peptide piece of the original protein that was represented by the chicken. And so the better your individual cell in your body can present all the different chicken parts on its surface, right, the more options you have for selecting for different T cells that can help recognize the chicken, right? Because each T cell will recognize either the chicken leg or the chicken wing or the chicken beak. So if you have an assortment of different MHC shapes that each can present a different part or a different peptide, well, then you can have a stronger immune response, right? So what we are saying then is because different shaped MHC platforms, right, would do a better or worse job of presenting specific peptides, right, if those peptides end up being the thing that the T cells mistakenly identify as being a threat, leading to autoimmune reactions, then your HLA polymorphisms then would sort of have an influence as to whether or not your cells end up presenting this peptide a lot, or if your cells end up not being able to present this peptide, right? And that would then translate into your risk of your immune system potentially making a mistake and seeing that peptide and leading to autoimmune disease. All right. So remember that these polymorphisms are given these types of labels. Like, for example, if you look over here, HLA-A asterisk 01:01, right? That's how they catalog all the different genetic variations. They have these different codes. What this figure on the right is showing, again, right, is that, and remember, these HLA genes are kind of grouped together on the same stretch of chromosome known as our MHC complex. And so that means when these polymorphisms are passed on and inherited, right, from parent to offspring, these individual genes aren't just randomly, right, reassembled and resorted. You tend to inherit chunks of DNA. So this chunk of DNA that would happen to have this set of different polymorphisms for HLA-A, HLA-B, C, and these various HLA-D genes, right, that code for your MHC2 shapes, will travel together. Therefore, they can be given, right, sort of an overall sort of label as this particular combination of polymorphisms, right, happen to travel together through inheritance from one generation to the next. And if this particular combination of HLA genes leads to specific shaped MHCs that might cause problems in terms of presenting peptides that the immune system has an easy time making a mistake against, then we can say that your risk of developing a particular autoimmune disease, right, can actually be either dramatically increased or maybe even reduced, depending upon what this haplotype block is that you carry, right, from mom and dad. So here you can see that on the left are a table of various different autoimmune conditions. Some of these you may have heard of, some of them you may not have. For example, there are things like type 1 diabetes here, right? There are things like rheumatoid arthritis and celiac disease, right? So these are conditions that we now know there is an associated risk, right, if you have certain inherited haplotypes, certain versions of these HLA polymorphisms. Okay. So what I wanted to kind of end with today is an example of one of these, celiac disease, right? So celiac disease, importantly, is not a true autoimmune disease, right? Now, what do I mean it's not a true autoimmune disease, right? So celiac disease is when patients develop a sensitivity to gluten, right? So this is what's often called gluten allergies, right? So what happens is that gluten is a protein that's very abundant in wheat and other grains. And this protein, right, has a lot of glutamines in it and glutamates, those amino acids. Because of the charges on these amino acids, when the gluten protein ends up being digested and processed by the cells of, for example, the GI tract, right, the little peptides that are sort of generated from the gluten have certain charges to them, right, that make it kind of tricky for certain shaped MHC platforms on the surface of the cell to hold on to and present. So certain shaped MHC platforms are very good at hanging on to and presenting these gluten peptides, right? And that means people who have those particular shaped MHCs, their cells of their, for example, intestines will present a lot more of these gluten peptides for the immune system to check out, compared to other people, maybe whose MHC shapes do a lousy job of hanging on to and presenting the peptide, right? Therefore, in those people, maybe their immune system never even gets to see these peptides, right? And if this peptide ends up being the target of a hypersensitivity, in other words, T cells that maybe should not have been allowed to go to work, right? They end up seeing these gluten peptides and causing an immune reaction. Then you can see, right, depending upon how your shaped MHC platforms are, that might influence your risk of actually this mistake happening. So you can see here on this table that the genetic component of celiac disease or gluten allergy has been traced back to these particular haplotype blocks, right? So these are the HLA polymorphisms that can modify or alter your risk of actually becoming gluten intolerant, right? So you'll notice that if you happen to have inherited these particular genetic variations, right, your risk of developing gluten intolerance is high, right? If you inherited maybe these particular combinations of those variations, maybe you're considered medium risk for developing gluten allergy, right? And interestingly, if your genetic variations are different than these up here, then you are at very low risk of developing gluten intolerance, right? Again, simply because the likelihood of T cells making a mistake and targeting the cells of your intestines, right, that present these gluten peptides, right, that all has to do with whether the cells are even presenting the peptide in the first place, right? So you can see here down below, here's the normal epithelial makeup of the small intestines, right? Somebody who's suffering from gluten intolerance, their adaptive immune system begins to attack and destroy their intestinal lining, right? And that can lead to conditions such as gastrointestinal problems, indigestion, diarrhea, a lot of discomfort, etc., right? So why is this not a true autoimmune disease? It's not a true autoimmune disease because the particular peptide here that is causing the problem does not actually originate from your own body as a self-signal, right? It comes from the food you are eating, right? It is coming from the gluten in the wheat, in the bread, for example, right? So somebody who's diagnosed with gluten intolerance, there's a very simple treatment for that. You just simply avoid eating foods that have gluten in it, right? Because if you don't get the gluten in your body, your intestinal cells won't ever display gluten peptides, and you won't have this autoimmune reaction. So that's why celiac disease, even though it is autoimmune in the sense that it's your T cells attacking your own intestinal cells, right? It actually fits more of a description of an allergy, right? Because the actual antigen that's being targeted here, right, is coming from an external source, right? So that's why people who have gluten insensitivity or gluten intolerance, they can just learn to avoid gluten-containing foods, right? But again, over the last couple of decades, we've seen a steady rise in the number of people who have gluten intolerance, right? And just like with allergies, because again, this is technically more of like an allergy, right? The question of how this happens can be traced back to our discussion about the hygiene hypothesis, right? Leading to more hypersensitive types of individuals in our modern society. Okay, so that's the end of today's lecture. All right, so I hope you find this discussion interesting. So we'll continue talking about this in our next class. All right, so for now, all right, bye-bye. Have a good day.